Cell F5FU. What is fluorouracil? Fluorouracil is a chemotherapy drug called an anti-metabolite that mimics that is one of the building blocks of RNA and DNA, which disrupts the ability of the cells to make DNA proteins. Fluorouracil slows or stops the growth of cancer cells and dividing cells, killing them. It treats a number of different cancer types, such as breast, colon, pancreatic, skin, and stomach cancer. A few other names that this chemotherapy drug is known as Carac, Efudex, Fluoroplex, and Tolac. As you can see, this is the structure of F fluorouracil, which is C4H3FN2O2. How does this chemotherapy drug works? This drug works through indigestion or topical application. 5FU enters cells through facilitated transport mechanism, which is converted to fluorodeoxyuridine monophosphate, which is also known as FDUNP. FDUNP plus diamidylate synthase, also known as TS. This inhibits the production of deoxythymidine monophosphate, which is also known as DTMP, which is essential for DNA replication and repair. Without DTMP, an imbalance of intracellular nucleotides leads to double-stranded breaks in the DNA by the enzyme endonuclease. This drug also serves as a pyrimidine analog by misincorporating into RNA and DNA in place of uracil or thymine, resulting in cell death of rapidly proliferating cells. The mechanism for the topical use of this drug uses a cytotoxic effect on actinic skin. Other mechanisms of this drug would be the interference with RNA processing and increase in PF3 expression. This treatment would be for actinic keratosis and squamous cell carcinoma. As you can see, this is another structure of the drug. How it affects RNA synthesis. L5FU is a nucleobase analog of uracil in which the hydrogen at position 5 is replaced by fluorine. It is a pyrimidine antagonist that functions by inhibiting DNA synthesis. F5FDUNP is an irreversible inhibitor of TS and hence of DNA synthesis through deoxythymidine triphosphate, which is DTTP. This deprives, which leads into apoptosis. As 5FU is similar in shape but does not undergo the same chemistry as uracil, it inhibits RNA replication enzymes, which therefore blocks the RNA synthesis and stopping the growth of cancerous cells. TS folate dependent enzyme. It also is a TS is a folate dependent enzyme that functions as an RNA binding protein. The S binds with high affinity to its own mRNA, resulting in translation repression. Research has shown that TS interacts with the mRNAs of the CMYC oncogene and the p53 tumor suppressor gene. These two genes have been shown to play critical roles in cell cycle control, DNA biosynthesis, and apoptosis. The, mis- the misincorporated of TS into the RNA is evidence suggests that RNA-based effects plays a significant role in its cytotoxicity. Not only does it inhibit the processing of pre-rRNA into mature rRNA, but also disrupting post-transcriptional modification of tRNAs and the assembly activity of snRNA, which or protein complexes, thereby inhibiting splicing of pre-mRNA. Some side effects of fluorouracil would be diarrhea, vomiting, nausea, and dehydration. Some serious side effects would be neurotropenia, pyrexia, pulmonary embolism, thrombocytopenia, and leukopenia. Now, these side effects can affect 10 in 100 people, more than 10%, and for the more serious side effects, it would affect, I believe, 1 in 100 of people. This is the metabolism of 5-FU, just showing you. This is two different structures, giving you two different ways to see how 5-FU works in the cell. As you can see, 5-FU, when it goes to FDUNP, it runs into TS, which the DNTP imbalances increase DUTP, which gives you DNA damage, which affects the p53 gene. When you go to the other side, 5-FU goes to FUTP. With the help of uridine, it also damages the RNA, which affects the p53 gene. This is another figure at the bottom, just showing you another way of it. And here you go. You got the citations here. And thank you.